Hello guys, my name is Kirtan and welcome to my channel Physiology at Once. Today I will be talking about the topic white blood cells. It is one of the most important topic in the blood physiology unit, and the most frequently asked questions from this topic is the neutrophil phagocytosis process, which I will be talking later in this video. So let's start. The white blood cells participate in the defense functions of the body, and the total leukocyte count in an adult is 4000 to 11,000 per millimeter cube of blood. It is the normal value. The white blood cells can be divided into granulocytes and agranulocytes. Under the granulocytes there are neutrophils, eosinophils and basophils, and under the agranulocytes there are monocytes and phagocytes or lymphocytes. There are three phases in the life of WBCs: marrow phase, circulatory phase and tissue phase. The most of the leukocytes stay 4 to 6 hours in the circulatory phase and almost 4 to 6 days in the tissue phase. The monocyte, which is one of the white blood cells, converts into macrophages in the tissue phase of the life. Count. Neutrophils contain 50 to 70% of all WBCs. Eosinophils contain 1 to 4% of all WBCs. Basophils contain 0 to 1% of all WBCs. Lymphocytes contain 20 to 40% of all WBCs. And monocytes contain 2 to 8% of all WBCs. So from this we can say that neutrophils contain the major amount of WBCs of the total WBCs, and basophils are the least. So now let's talk about the neutrophil. As discussed, it is the most common WBC in the peripheral circulation, and that is 50 to 70% of WBCs. They act mainly against acute pyogenic infections. The structures of the neutrophils. The average size is 10 to 14 micrometer, and the cytoplasm contains fine pink colored granules. They are multi-lobed nucleus. It may also contain the bar body in the nucleus. The neutrophils can be further divided based on the number of nuclear lobes, known as the Arnett count. Now let's talk about the functions of the neutrophils. They are actively phagocytic. They contain many antimicrobial and bactericidal chemicals in their granules. They are the first line of defense against acute bacterial infections, and they also play the role of major non-specific defense against the invasion of pyogenic organisms. Therefore, the decrease in the number of neutrophils, which is also known as neutropenia, predisposes the body to pyogenic infection. Now let's talk about the most frequently asked question from this topic, neutrophil phagocytosis. It is the most important question. It is a process of ingestion and killing of microbes or foreign substance by a phagocyte, which is nothing but the neutrophil in this case. There are 5 steps in this process: chemotaxis, diapedesis, optionalization and adherence, ingestion and killing. So let's talk about these steps one by one. Chemotaxis. It is the process of migration of neutrophils to the site of infection. The chemical substances are released from the inflammatory cells, which act as attractants of neutrophils to the site of infection. These are called chemotaxin. So many chemical substances act as attractants to attract the neutrophils towards it. So neutrophils comes towards it, and this process is called chemotaxis. The main attractants in the body are complement proteins, that is C5a and C3, and the leukopoiesis is stimulated on increased demand of neutrophils. So if the infection is more, the neutrophils demand will be more and the leukopoiesis is stimulated. The next step is diapedesis. It is the process of transport of neutrophils through the capillary endothelial cells to reach the invader in the tissue. Since the neutrophils are present in the blood, it needs to be transported to the tissue through the capillary endothelial cells. So this is done through the process of diapedesis. The activated neutrophils first marginate and adhere tightly to the endothelial lining with the help of L-selectins. So first, neutrophils adheres to the margins of the blood vessels. First it will be flowing in the center, but then after in the process of diabetes, it goes to the margins and adheres to the margins of the capillary tissues and then passes through the capillary tissues in the process of diabetes. Then by their amoeboid movement, they squeeze through the space between endothelial cells to reach the tissue. The next step is opsonization and adherence. The bacteria should be made tasty to the phagocyte for the neutrophil to attack it. So it is done by many opsonins, which are immunoglobulin G and C5a and C3, which are nothing but the complement proteins. Complement proteins. The attachment of phagocyte to the membrane of microbe is called as adherence. The next step is ingestion or endocytosis. In this, the phagocyte encroaches the microbe by its projection called pseudopodia. They are the false foot of the neutrophil, which spread across the microbe and ingest it. The pseudopodia surrounds the microbe and forms phagocytic vesicle. This phagocytic vesicle fuses with the lysosome to form the phagolysosome. The last step is the killing step. It is also divided into two types, non-oxidative and oxidative. In the non-oxidative mechanism, the antibacterial chemicals produced from the neutrophil granules are the main killing factors. The antibacterial chemicals. Some examples are lysozyme, which hydrolyzes the cell wall bacteria, lactoferrin, which sequesters the iron. Sequesters means it blocks the iron, which is the main required factor for the growth of bacteria. Defensins, it disrupts the outer membrane of bacteria and breaks the single-stranded DNA structure, due to which the bacteria cannot further divide and is killed. In the oxidative mechanism, the oxygen metabolites produced from the activated neutrophils are the main killing factor. The oxygen metabolites. So some example are superoxide anion, H2O2, free hydroxyl radicals, hypochlorous acid, and singlet oxygen. The superoxide anion is the most effective oxidant metabolite. The activation of NADPH oxidase is associated with increased oxygen intake of neutrophil. This process is also called respiratory burst. This process is required for the production of superoxide anion, which is the most important oxygen metabolite in the oxidative mechanism. The process is NADPH plus H plus 2O2 gives rise to NADP plus 2H plus 2 superoxide anion. This is an important process and you need to remember this. The next example is myeloperoxidase. It facilitates the conversion of chlorine minus to HOCl, which is also a potent oxidant. There are many applied aspects in this case. The chronic granulomatous disease, which is short formly called CGD, occurs due to defective NADPH oxidase. In this case, the NADPH oxidase is reduced and due to which the superoxide anion cannot be produced in adequate amounts. And this leads to ineffective oxidative mechanism and ineffective neutrophil phagocytosis, because oxidative mechanism is one of the main mechanism in the killing process. Neutrophil hypomotility. Neutrophil needs to be mobile, due to which it can go to the pathogen and kill it. The primary phagocytic dysfunction of neutrophil is the neutrophil hypomotility. It decreases the phagocytic activity because the neutrophil is no more motile. The interaction of oxidative and non-oxidative mechanisms. They run both hand in hand, due to which the process is enhanced. The non-oxidative mechanism facilitate the activity of oxidative mechanism for bacterial killing. The proteases and the collagenase enzymes produce a killing zone around activated neutrophil by locally damaging the inflammatory tissues. So we can understand this like there is an activated neutrophil and this is the surrounding tissues, and the killing zone is produced across the activated neutrophil by the proteases and collagenase enzyme. This is the killing zone, and in this zone the oxygen metabolites are more and the oxidative mechanism will be enhanced in this zone. So the oxidative mechanism will be increased. This helps the oxidant molecules to promote their bactericidal properties. So they run both hand in hand and enhance the process of killing. So let's talk about the next WBC, which is eosinophil. It forms 1 to 4% of all WBCs and it is 10 to 14 micrometer in diameter. It has bilobe nucleus and has coarse brick red granules in its cytoplasm. It has short lifespan in the circulation phase, whereas longer in the tissues. It is mainly present in the epithelia of respiratory, GIT and genitourinary tract. Their production and function are mainly influenced by the interleukin 5. I talked about this factor in the video leukopoiesis, in which I told the interleukin 5 enhances the process of the production of eosinophil. Their functions: they participate in two important defense mechanisms, in helminthic infections and in allergic infections. In the helminthic infections, they attack the intestinal parasite by the process of first eosinophil attach themselves to the opsonized larva and get activated. Then within 3 hours, the activated ones secrete the proteins. Then it secretes toxic mediators that destroy the tissues of larva. And in the allergical reaction, like bronchial asthma, it plays the major role. So oftenly it is said that in the bronchial hyperactivity, it is always correlated with the eosinophilia and airway eosinophil content. So when the eosinophil content is more, you can say that there is bronchial hyperactivity, and it is often decreased by giving the dose of glucocorticoid, which decreases the content of eosinophil in the body. The next WBC is the basophil. It is the least number of WBC of the total WBCs and forms 0 to 1% of all WBCs. It is also 10 to 14 micrometer in diameter. You must have noticed by now that both neutrophil, eosinophil and basophil have 10 to 14 micrometer in diameter size. It is bilobed or irregular nucleus, and it has deep blue-purple cytoplasmic granules. The mast cells resemble the basophils functionally but are not identical. They both are separate. The functions of the basophil are: they play a major role in acute and chronic allergic reactions. They also play a role in T cell dependent responses. T cell is nothing but the lymphocyte. It also plays a major role in host defense. The next WBC is the monocyte. It forms 2 to 8% of all WBCs. It is a bit larger than neutrophil, eosinophil and basophil. Its diameter is 12 to 25 micrometer. Its nucleus is oval shaped or horseshoe shaped. Its cytoplasm is turbid in appearance. The function of monocytes are: it helps in phagocytosis and microbial killing. It helps in the antigen presentation process, and it helps in the release of cytokines. So that's it for today, guys. If you found this video informative, please like, share and comment, and also don't forget to subscribe.